in last few lectures I have introduced you to 3D extrusion printing as one of the most promising additive manufacturing route to manufacture scaffolds of soft materials. So, in this lecture I'll share with you one of the scientific case study on Jelma hydrogels for hard tissue applications, and the manufacturing route employed here is 3D extrusion bioprinting. You may recall the hydrogel by definition, it is a macromolecular network structure of hydrophilic polymer with the ability to retain significant amount of water molecules. Okay. So, this was the basic definition of hydrogels. So, Jelma by far is one of the most widely investigated hydrophilic polymers which are used for the hydrogel fabrication in academic setting or in the research laboratory, and hopefully the clinical translation of these Jelma hydrogels will be for patient care will be realized in a much larger extent in the years to come. So, the process science is very important, and I have emphasized the process science for metal 3D printing. Now, this slide you might have you have seen it before, but to recapitulate, I thought that I will show and explain very briefly this slide once again. So, your hydrogel is loaded here in this nozzle, and it will experience pressure. So, when you use biomaterial ink, so this biomaterial ink will not contain cells, but when you will use bio ink, as you can see here, this is the bio ink formulation. When you will use bio ink, that will contain cells. So, one of the major challenges in the formulation of bio inks is that the cells which are contained in these particular soft hydrogel matrices should not undergo cell death, so that the cell viability is not compromised when encapsulated by hydrogels. Okay. So, this is the major things, and therefore, biomaterial ink formulation should have desired rheological properties in terms of viscosity, gel strength, shear thinning behavior, and post printing recovery like thixotropic properties and all. It should have good extrudability, and this extrudability while it is extruded under pressure, right, under pressure P, that one has to have a very stable long filament structure, and without any breakage, without any shape distortion of the filaments, and this can constantly and consistently will be printed on a support structure. And then third one is the buildability. Buildability is like, how this structure can be built with different infill design, and also layer heights, line width, line spacing, but most importantly I have mentioned multiple times and I am mentioning is again is the safe fidelity compliance. So, that is very important, safe fidelity compliance. And as I said that cell fate processes, it is not only viability but also functionality of the cells when encapsulated within the hydrogel, that is also equally important, and that has to be assessed or analyzed at different time scale. It is up to few weeks in culture, and one of the major thing in the tissue in the 3D bioprinting is the tissue maturation. And tissue maturation, that length scale of which you need to do the cell culture or 3D cell culture of the 3D printed hydrogels, that length scale can vary, like it can go up to few weeks in culture. Right. So, selection of biomaterials. So, these are like bioprintable materials. Biomaterials, you have a additive polymers like natural or synthetic, and out of this natural synthetic you have a PEG, polyethylene glycol, PVA, polyvinyl alcohol, polycaprolactone, PCl, then this is you have PEG, then this is you have PVA, and then you have a polylactic polyglycolic acid copolymer PLGA, then you have polyurethane PU, and there is pluronic F127. These are like different synthetic polymers, right, which can be 3D bioprinted. Then from the natural polymers you can have a decellularized extracellular matrix, that is DECM. You have polysaccharide based natural hydrogels, you have a protein based natural hydrogels, and there you will have collagen, gelatin, silk fibrin, and gelatin and matrigel. In the polysaccharide case you have cellulose, agarose, and then I will show you also in one of the case study where nanocellulose particles are added to one of the hydrogel ink or one of the biomaterial ink, that is alginate hydrogel. Then you have a gel gum, these are like carbo polysaccharide base, chitosan, alginate, hyaluronic acid. So, these are like some of the prominent polysaccharide based biopolymers. Then in the case of these other materials like in the mineral based inorganic fillers, you can have silicate based like bioactive glass, 4,5S5, nano clay, mesoporous silica nanoparticles. Then you will have calcium phosphate based materials like hydroxyapatite, tricalcium phosphate, and so on. In carbonaceous materials, these are like carbon nanofibers, graphene, oxide, carbon nanotube. Okay. So, this one is mineral based inorganic materials. These are like additives to the hydrogel bioink, which will impart specific properties to this biomaterial ink, right. Now, this side of this part, it is like this side of this, like it is like these are like again used either as a matrix component or as viscosity modifier and so on. So, that you will see in some of the in one of the scientific case study under the broad theme of the 3D extrusion printing. So, it should be clear from this slide. Let me clear it off what I have written. It should be clear from this slide. You in one side you have the major matrix components like collagen, gelatin, matrigel, silk fibroin. So, these are like protein based matrix component of the hydrogel. So, to recap what I have just discussed about the bioprintable biomaterials. So, I will put a star here just to emphasize that this protein based biomaterial ink is most commonly used like collagen, silk fibroin or gelatin, and it is all natural derived materials. So, then decellularized extracellular matrix, what is decellularized? If you recall, tissue is nothing but cell plus ECM, right. I mean, it is not a mathematics, but in a biological context, tissue essentially combines the cells, the group of cells with similar functionality which are dispersed or which are embedded in an extracellular matrix. So, what is the meaning of decellularized matrix? Decellularized matrix, extracellular matrix essentially means by certain chemical or biochemical processes, if you adapt to remove the cells from a tissue, like there is no cell, then what will remain from a tissue? It is decellularized ECM. Okay. Or if you grow cells over time, you allow cells to synthesize extracellular matrix. Then intelligently, if you remove the cells from the medium, then you will be left with that decellularized extracellular matrix. Either way, you start with the tissue, you adopt biochemical process, or you grow cells, you allow cells to synthesize extracellular matrix proteins. Then when it gets matured, then you intelligently remove the cells to get only decellularized matrix. So, this is also another scaffolding material. Then you have polysaccharide based materials, there is additive like cellulose, like nanocellulose, like chitosan, gelen gum, alginate, and then some of them are also can be used as a matrix component of the hydrogel biomaterial ink. So, these are the natural side, and synthetic side, as I said, PG, PVA, PLGA, PCl, they are all used. Yeah, and these part are essentially used for the additives like inorganic additives, either carbonaceous fillers or calcium phosphates or silicate based like bioactive glasses and so on. Right. So, as I said that gelatin, GELMA is perhaps the most widely investigated matrix component of the hydrogel ink. So, I should introduce you in more details about the overall GELMA synthesis and chemical cross linking of GELMA. So, what I was talking to you that there are three ways that GELMA can be synthesized starting from gelatin. One is a conventional method, one is a sequential method, and third one is a one pot facile method. Now, what you remember from the earlier discussion is that in the gelatin you have NH2 amide side group. Now, you want to treat them with methacrylic anhydride MA, and as a result that you produce GELMA and methacrylic acid. Now, all these synthesis technique, if you see, their reaction environment is different in terms of the conventional method. People use phosphate buffer saline at 50 degrees Celsius 1 to 6 hours, and in the sequential method, researchers use carbonate bicarbonate buffer 0.1 molar concentration 50 degrees Celsius for 3 hours, and there is a sequential pH control and then temperature, right. Then third one is that facile 1% synthesis, that pH adjustment and methacrylic anhydride addition, that is carefully controlled, and again carbonate bicarbonate buffer is used. Now, question is that why GELMA is so widely investigated. Now, if you look at this backbone chain of the GELMA, in this backbone chain, if you look at this red part that which has been zoomed here, it has the RGD motif. What is RGD? RGD is arginyl glycylyl asparic acid. So, this RGD is a very common peptide motif which is attached to which is responsible for cell adhesion to a ECM. Now, what is ECM? That I have already explained to you earlier, right, extracellular matrix. So, because of the presence of RGD motif, which is the 3 amino acids together, and this is a particular arginyl glycylasperic acid. So, this is a common peptide motif which is responsible for cell adhesion to extracellular matrix. Now, there are 3 ways you can do this physical gelation. One is the physical gelation. You can essentially treat them at a less than the at temperature less than the gel formation temperature. Then you can use the chemical cross linking. Essentially, you can add that IGA-Cure or some of the photo cross linker, expose them to UV. Then third one is the coordinated gelation and cross linking. So, essentially you give both, or you can mention is the dual cross linking, like you can do both UV based linking as well as the physical gelation. So, the storage modulus, that you will see that G prime, when you measure the rheological properties of these materials, the storage modulus keeps on increasing when you have more and more cross link density. So, certainly you can see if you go from here to here, that molar cross link density is expected to increase, and what you see, that is that this is a kind of a cell. So, this is a kind of fibroblast cells, and fibroblast cells are attached to this GELMA, and this is also another type of cell. So, this cell adhesion and cell spreading, what we are trying to show you here, that also increases because of the larger storage modulus of the GELMA. Now, key points to remember. So, I think I have been discussing with on this collagen and gelatin and GELMA for quite some time now. So, at this point it is instructive me to summarize some of the key points so that you can remember. So, gelatin is the denatured form of collagen, that you should always remember. And what is collagen? Collagen is the organic matrix of the natural bone. Now, GELMA is essentially derivative of gelatin, which has specific RGD motifs to attach biological cells, that also I have explained. GELMA as a primary matrix constituent for hydrogels because of better gelation property. GELMA as a primary matrix has been most widely investigated for tissue engineering and 3D bioprinting. Fourth one is that high concentration based GELMA, as you will see later, that has been more widely explored in the additive manufacturing community. And collagen is sourced from cartilages of fish, pig means porcine, or cow means bovine. Fish scale collagen or gelatin, fish scale collagen gelatin is much less investigated in the scientific community. Now, if you look at that historical evolution of GELMA over last 2 decades, if you start with this 2000, so GELMA was first introduced as a photo cross linkable polymer. Then subsequently in 2010, this is the first used first used as the cell encapsulated GELMA microgel, right. Then 2011 scientists have shown that GELMA can be also micro patterned of cell laden GELMA bioink. Then cartilaginous matrix bioprinting was reported in 2013, then goes on series. So, as you see that from 2010 onwards, the intensity of research on GELMA has also increased, right. And if you see that more number of papers started coming, like for bone tissue engineering, JELMA was shown to promote vascularization. Then dental bioprinting was reported in 2015, then goes 2016, several papers like JELMA has been started, JELMA has been investigated for wide spectrum of artificial organs, for example nerve tissue, cardiac tissue bioprinting, liver constructs, cancer at drug delivery. Then JELMA started used for the microfluidics and stereolithography, skin bioprinting and so on. Then 2022 scientists have reported also for 3D bioprinting of liver constructs using JELMA. 2021 there is a paper for JELMA of the bioprinting of artificial ovaries. So, as you see that the research on JELMA, scientific research on JELMA in different research groups around the world has really intensified over last 2 decades. So, for example, that JELMA foam has been used for this JELMA foam and hydroxyapatite, this combination was used for bone tissue engineering. I will give such an example. So, if you look the natural bone has the outer cover, that is the cortical bone, this is the harder part of the bone. Then you have a cancellous structure which is the inner part, this is spongy and porous, and then you have the trabecule, right. So, natural bone you have a lamella, and this lamella is made up of the collagen fibers, and if you go down to the structural details, so collagen fiber contains tropocollagen and hydroxyapatite crystals. Now, hydroxyapatite can be synthesized by different methods. So, one of the methods which uses the nitrate precursor, for example calcium nitrate 4H2O or ammonium biphosphate, this one. So, this where calcium to phosphorus ratio is typically maintained at 1.67 for the stoichiometric HA, this is the autoclave based synthesis route. So, this autoclave based synthesis route, one of my former student Soumitra during his PhD at ISC, he has prepared and showed that how hydroxyapatite needle shaped can be formed by these methods. Now, JELMA synthesis, as I have shown you before, you start with the PBS buffer 50 to 3 hours. This is a conventional synthesis technique that we follow in our group, then dialysis, deionized water for 7 days, then lyophilization, then JELMA foam is prepared. Now, degree of substitutions in the case of JELMA is typically determined by NMR spectroscopy, and NMR spectroscopy you have when the JELMA is formed, then you will get the signature NMR bands H and HB and HY and HX, which normally you do not see in the gelatin, right. So, now if you look at these particular things, so it is the what is the lysine methylene peak area in JELMA and what is the lysine methylene peak area in gelatin, that ratio that you have to subtract from 1, then you get this degree of substitution. As I said just 2 minutes back, this autoclave based synthesis route which was pursued in my group, and then they found that what is the morphological analysis, size distribution, anisotropy of JELMA. Now, what you can notice here that if the reaction time, so in this particular process we have changed both the reaction time and then reaction temperature. So, reaction temperature at 200 degrees Celsius, you can see very nice needles, and these needles has an aspect ratio, and this aspect ratio C by A has been at least 8 to 10, and you can see here that what is the particle diameter. Particle diameter is roughly very small, it is like nanoscale particle diameter, like 30 to 40 nanometer. So, phase pure anisotropic needle shaped hydroxyapatite, these were utilized as nanofillers in JELMA in regulating biophysical properties and printability. Now, what you see here, that scaffolds buildability. So, first I will show that 15% JELMA which was used initially as part of Soumitra's PhD thesis. Now, if you see that 15% JELMA, when it is being printed, you cannot have a good structure, or structural fidelity or shape fidelity certainly is not maintained, right. Now, if you go to 15% JELMA with 1% hydroxyapatite, that nanoscale hydroxyapatite, you do see that structural resolution and structure is being built. And when you add this 3% GELMA, 3% hydroxyapatite to 15% GELMA, and you can see that this particular structure is being built very nicely, right. So, you can essentially see very nice structure. So, if you for the moment, if you just stop it, if you stop this movie and just if you concentrate on this movie only, on this video only, you can see that how this 3% hydroxyapatite addition improves the buildability of this particular GELMA construct. Right. Now, limitation of the high concentration GELMA, for example, people have reported that NiH3 cell viability is actually goes on reduction with more higher percentage of the GELMA. If you look at embryonic stem cell proliferations, and if you look at this 3, 5 and 10% GELMA, so this cell viability also reduced, particularly after 6 days of implantation. Encapsulated, when you encapsulate MSC, mesenchymal stem cells, and then you grow them day 2, day 4 and day 6, then day 6 also this particular cell number decreases. Now, with GELMA, with larger concentration GELMA, there are reports from a Chinese group in bioelectrochemistry. There also you see that it goes below 74% or below 80% at 15% GELMA. So, therefore, high concentration GELMA, there are certain limitations, and that was the reason that why we have started working on the lower concentration GELMA. So, a lower concentration GELMA is preferable for maintaining optimal encapsulated cellular functionalities, and besides higher matrix stiffness, cross-linking density can potentially impose physical stress on embedded cells, while reduced matrix porosity limit nutrient supply. So, you need to have a perfect balance between the matrix stiffness as well as the porosity. So, if your matrix stiffness is higher and cross-linking density is higher, then what would happen? That can impose physical stresses on the embedded cells, that I am going to discuss with you at some point in this course. While reduced matrix porosity, suppose matrix porosity is reduced to improve the mechanical properties, then that will certainly reduce the nutrient supply, and then also vascularization process will be impaired. So, we have used in the low concentration GELMA, and low concentration GELMA we have used, but you have to modify the hydrogel composition, right. So, we have used carboxymethyl cellulose as one of the viscosity modifier, and if you see GELMA with carboxymethyl cellulose, and you cannot use straightway carboxymethyl cellulose. For example, if you do GELMA solution, if you do physical gelation, then you get uncross-linked GELMA gel. Then if you do UV cross-linking in the presence of photoinitiator, then you get physically cross-linked or these UV cross-linked GELMA hydrogels. Now, there is other way. Suppose if you use the carboxymethyl cellulose, but not pure carboxymethyl, but you modify so that it gets attached to this GELMA chain, then if you add this GELMA CMC solution, physical gelation almost less than 21 degrees Celsius, then you get cross-linked GELMA hydrogel, cross-linked GELMA CMC hydrogel. As I said that we are concentrating on the low concentration of GELMA, that was a challenge that we took. The 5% GELMA concentration that you can see in this particular data, if you modify it with 0.5% carboxymethyl cellulose, 1% carboxymethyl cellulose, 2% carboxymethyl cellulose, then suddenly you see the degree of cross-linking almost linearly reduced, right. So, degree of cross-linking goes down to almost like 60% at the 2% carboxymethyl cellulose modified or cross-linked GELMA hydrogel. So, There is also degree of cross linking that is measured at PBS 37 degrees Celsius. We have also measured degree of cross linking at at 2 days and then with different concentrations. So, there are 3 approaches that we have used in this UV cross linked modified carboxymethyl cellulose. So, what we have shown in this particular case, when we have done modification of carboxymethyl cellulose and then we have used as a viscosity modifier to GELMA, the cross linking degree of cross linking almost remains same. It is like almost like 95% or so. So, how these methacrylic modifications were done? So, you take this carboxymethyl cellulose, like in the, you remember gelatin to GELMA, you added this methacrylic anhydride, right. Now, you start with the carboxymethyl cellulose, you add this methacrylic anhydride and deionized water pH 8, 9 and in this ice conditions 0 degree Celsius 48 hours, you essentially add this particular group to this chain. So, then you make this methacrylate carboxymethyl cellulose, right. So, so, now if you see that 5% GELMA with carboxymethyl cellulose without any modification to CMC, you get this shape is certainly not maintained, right. So, when you add this 1% carboxymethyl cellulose, so you can see it is a good gel strength. Now, you add the 2% carboxymethyl cellulose, modified carboxymethyl cellulose to 5% GELMA, it is perfect, right. So, you can see the evidences that what these methacrylic modifications to CMC can do to improve the good gel strength and good gel properties, particularly in the case of this GELMA. And you can see the signatures of this methacrylation to carboxymethyl cellulose by NMR spectroscopy analysis. So, as you can see, this particular technique is quite extensively used to find out the signatures of any chemical modifications in this macromolecular structure. This is the FTIR. So, FTIR essentially see that again you can see this particular area which is like OH minus, but you can see there are also signatures of these other IR bands which essentially confirm these modifications, carboxymethyl cellulose modifications is done. So, this is like more chemistry based approaches. So, essentially what you see that when you start with this methacrylic modified carboxymethyl cellulose, then if you do UV cross linking. So, what would happen if you look at these particular bonds, you would see that this becomes more like a single bond, right. So, you get 2 modified carboxymethyl cellulose being bonded. Now, if you add that to UV cross linked and then expose this gel to UV cross linking, then essentially MCMC is now added to this backbone chain, right. So, this has been confirmed using chemical various spectroscopic techniques as well. In the next class, I will be essentially describing more on the properties as well as the cell compatibility of this particular GELMA system. Thank you.